Assalamualaikum. I'm Qurat from the Department of Fields Biotechnology, and today the topic of our presentation is thin layer chromatography. Thin layer chromatography is a technique which is used to separate the non-volatile mixtures on the basis of polarity. Principle: different compounds have different solubilities and adsorption to the two phases between which they are to be partitioned. Basically, it contains two phases: the stationary phase and the mobile phase. Here, a question is, why we use thin layer chromatography? The answer is to determine the number of compounds in a mixture and to determine the identity of two substances. Now we will discuss about the material which is required for thin layer chromatography. Number 1, glass slide, which is coated with silica gel, known as stationary phase. Number 2, tank, beaker or a chamber containing the solvent solution known as mobile phase. Number 3, sample to be identified. Number 4, pencil, fiber pencils. Number 5, cover of a tank or beaker or chamber. And number 6, burner, if we require. Now moving towards the method of the TLC, firstly we will take a glass slide. The glass slide may be ready-made or we can prepare in the lab. It's simple by just taking a glass slide and apply a thin layer of silica gel over it. But on the other hand, the ready-made glass slide I prefer better because the glass base is replaced with the aluminium base and the silica gel is uniformly distributed, which will be beneficial for the accuracy in the results. But the thickness of the glass slide may be microscopic or it may be bigger in size depending upon the sample which we are going to analyze. Then we will draw a line with a carbon pencil 1 centimeter away from the bottom of the slide and then apply the sample on the marked line in the form of a spot. Place the TLC plate in the chamber already containing the solvent solution in such a way that the marked line doesn't touch with the solvent solution. Because if the marked line touches to the solvent solution, the sample we have applied on the marked line will be mixed into the solvent solution and the results will not be predictable. That's why there must be a distance in the solvent solution and the marked line. Then cover the chamber, but the chamber should be saturated 20 minutes before we place the TLC plate into the chamber because the solvent solution contains vapors and the vapors must be evenly distributed and then it will be beneficial for the results. After some time, the solvent solution along with sample will spread over the TLC plate via the capillary reaction. The sample separated. Then the sample separated on the basis of polarity. This is the main important step because the polarity determines how the sample should be separated. On the basis of the molecules which are polar will move less on the silica plate because silica gel is itself polar. On the other hand, the molecules which are less polar will move more upward because of the solvent system. So that's why the polarity is the most important in determination of the components. Then we will remove the plate from the tank and measure the RF factor, which is the retardation factor. Retardation factor or RF factor is the ratio between the distance traveled by the component to the distance traveled by the solvent. So after determining the RF factor, we will compare the values with the reference, so identify the components. Now, the most important thing is visualize the sample. The sample may be colorless or colored. If the sample is colored, so it can be visualized with the naked eye. But if the sample is colorless, there are the 5 important methods which we use for the visualization of a sample. Number 1, UV visualization, which is used for the polyphenol type components. Acid treatment for the acidic type of components. Iodine vapor treatment for the identification of the double bonds. Valine stain, which is used for the alcohol, aldehyde, and ketones. And the number 5th or last one is Nin-hydrin test, which is used for the amino acids. So these are the 5 methods which are used for the visualization of a sample. Now we will discuss there are the 2 main important things about the spots. The spots doesn't occur, means the sample is applied in less amount, or if the strikes are formed on the plate, means the sample is overloaded. So these things must be keep in mind. The sample will not be applied in very large amount or in very less amount. That there is the problem in the end result. So there must be the spot must be applied in accurate amount so that the result will be predictable. Now moving on towards the applications of the TLC. There are many applications of TLC. Some of them are listed here. Number one, the qualitative testing of various medicines such as sedatives, local anesthetics, antihistamines, steroids is done by TLC. TLC is extremely useful in biochemical analysis such as separation or isolation of biochemical metabolites from its blood plasma, urine, blood, body fluids, serum, etc. Thin chromatography can be used to identify natural products like essential oils or volatile oil, fixed oil, glycosides, waxes, alkaloids, etc. It is widely used in separating the multi-component pharmaceutical formulations. It is used to verify of any sample and direct comparison is done between the sample and the authentic sample. It is used in the food industry to separate and identify colors as a sweetening agent and preservatives. It is used in the cosmetic industry and it is also used to study if a reaction is complete or not. What are the disadvantages? Thin layer chromatography plates do not have longer stationary phase. When compared to other chromatographic techniques, the length of separation is limited. That's why the results generated from the TLC technique are difficult to reproduce. Since TLC operates as an open system, some factors such as humidity and temperature can be consequences to the final outcome of the chromatogram. The detection limit is high, and therefore if you want a lower detection limit, you cannot use the TLC technique. It is only qualitative analysis technique, not the quantitative. These are the few disadvantages on the TLC technique. That's all from my side. Thank you.